get started. Yeah, and well, first, welcome everybody to the MRSEC seminar, and this is a monthly seminar series for the newly established MRSEC Center and from NSF. So today is my great pleasure to introduce speaker, Professor Joop Boekhoven from Technical University of Munich. I try to pronounce your name right. We'll see. So thank you for taking the time, and particularly this is Friday evening, so for German local time. So this is before the holiday season and Friday evening. Thank you for taking the precious time sharing your science with us. And let me give a brief introduction. And we just chatted before this got started. He has quite a bit of international experience. He was born in Brunei in Southeast Asia. And when he was 4 years old, he moved back to Europe. He lived in a few countries in Scotland and then Holland and then the Netherlands. But his formal education, higher education, seems like all from Netherlands. And his bachelor degree, master degree from University of Groningen, and then his PhD from TU Delft. And he worked under one professor for 2 institutes, but the same gentleman, Professor Jan Vanesh, and including his PhD dissertation work. So at that time, Joop started working in this new field, chemical field dissipative assembly. So which Joop called version 2 for self-assembly. So which I look forward to his story. And after he finished his PhD in 2012, he came to the States and with Rubicon postdoctoral fellowship and work with Sam Stoop at Northwestern. And naturally, he worked on peptide assembly. So that's Sam's I think a real passion, and for biological application as well. I think I just saw another paper from his group published in Science for spinal cord repairing. So I don't know which project you worked on. Yeah, and then so in 2016, after 4 years in the US, and he returned to Europe and started his independent career at Technical University in Munich. And he joined as a Moosbauer assistant professor chair, and which Munich is well known for the chemistry program for the science. And since then, he established a highly successful and highly visible, exciting research program. And so Joop come to the scene as one of the real pioneers in the new field for this dissipative self-assembly with chemical reaction as a fuel. So he attracted my attention, I think, from reading your paper, 2015 science paper. So Joop co-authored with Vanesh, but Joop was the first author for this paper, assuming majority of the work was carried out by Joop. So that was really a landmark and a milestone paper for the field, and is highly cited and highly recognized, because that really opens the new avenue and chemical reaction to fuel self-assembly, which he continued pioneering this field in his independent career at TU Munich, and developed a number of new exciting directions. So today, we look forward to hearing it. And before I let you start, I think Joop, for his scholarship and also contribution, he won a number of awards, and at his very young early career. And just name a few, he received the ERC starting grants, which is highly competitive, and he was elected Max Planck Fellow twice. He also received the Volkswagen Life Fellowship, and most recently, he received his VCR Lecture Award from the German Fund of the Chemical Industry. So I really enjoy your science. I look forward to your seminar. Okay.
Fantastic. Thank you very much, Steven. Thank you for that kind introduction, and also thank you for having me here. Looking very much forward to talking to you about what we've been doing in the last years. It's a shame I cannot make it to California. Would have enjoyed the weather. It's probably better than we have over here, but maybe in the future. What I'll be talking about today is molecular self-assembly, which I'm sure you're all familiar with, but then regulated through the use of chemical reactions. And we've been working on this in order to create a toolbox towards new materials, models for biology, and perhaps one day the synthesis of life. This is an example of the peptide amphifiles that I worked with in Sam Stoop's lab. And I use this as a demonstration for molecular self-assembly, which I think most of you will be familiar with, where we're looking at molecules that are designed to interact with each other through non-covalent interactions. So we're talking about hydrogen bonding, hydrophobic interactions, etc. And as a consequence of these non-covalent interaction, we create a larger ensemble, and the product, the molecular assembly, has properties which is greater than the sum of its parts. So we can now design molecules that assemble, and then the assembly has different properties than the individual molecule would have if it were not to assemble. Peptide amphiphile is one example. A more relevant examples, particularly these days, are the lipid nanoparticles that are used for delivery of mRNA, for example, in the vaccines that have been widely distributed. So in this case, we have lipids that come together through hydrophobic interactions and ionic interactions. And the role of these lipids in this case in their ensemble is to encapsulate mRNA and then protect this mRNA from degradation through water or hydrolysis or degradation by pH, nucleases, oxygen. So this is a beautiful example of where molecular self-assembly is applied as a toolbox for materials. And I think if we now look at life, the cell, for example, is also a molecular assembly. However, this molecular assembly, as you can already see in this slide, is different than the classical assemblies that we're used to. It's more dynamic. It is moving around. In this case, what you're looking at is the filaments within the cytoskeleton that are building up and rearranging in order to perform its dynamic functions. And if I look at this and I classify myself as a chemist, then I see the structures in these cells. The folding of protein, the cytoskeleton are also formed through self-assembly, the self-assembly that we are familiar with, that we're good at as supramolecular chemists. However, the function of these assemblies is typically regulated through chemical reactions. So you can see the rearrangement of proteins or the refolding of proteins or the assembly of the cytoskeletal network is typically regulated through chemical reactions. And that's something we are not very familiar with in this field of self-assembly and supramolecular chemistry. So we've been wondering, can we perform similar functions if we would apply chemical reactions to our molecular assemblies? This may be a little bit vague, so let me give you a nice example from biology where function is regulated through chemical reaction cycles. And this is a catalytic reaction cycle in which we are effectively hydrolyzing one molecule of ATP. So this is the chemical reaction that we're running. ATP plus water gives us ADP plus inorganic phosphate. This reaction is extremely slow in water. It has ATP has a half-life in the range of thousands of years. So we need a catalyst for that. And the catalyst is our molecular assembly. In this case, we're talking about calcium 2+ ATPase, and the ATPase already gives away that is our catalyst that is going to hydrolyze our ATP. And that's sitting in this case over here. It's a folded protein that is a transmembrane protein. So it's spanning a membrane that's drawn in yellow over here. So we have the membrane over here that separates the cytosol from the inner lumen. And what we're doing over here is we're pumping calcium from one side to the other side. We're pumping it against a calcium gradient. So this is definitely energy unfavored. You need to invest a little bit of energy to get calcium from there to there because the concentration of calcium on the other side is higher. That energy is going to come from our ATP. And the way this works is that we have two ions of calcium combine the protein. This can now react with ATP, and upon reaction with ATP, you could say it gets activated. It undergoes several morphological transitions. The protein refolds, and by refolding it, it pushes the calcium to the other side, where it can be released into the other side, right, into the other aqueous phase on the other side of the membrane. Importantly, the phosphate is attached, and in the last step, the phosphate gets cleaved off. Now the reaction is complete. We have ATP plus water gives ADP plus phosphate. And the molecule goes back to the original state. So it's a catalytic reaction cycle. This was merely a catalyst, which can now undergo a new cycle and a new cycle and a new cycle. It can do this hundreds of thousands of times. And what we're effectively doing is we're using this catalyst to convert ATP into ADP. And the energy that we are gaining through that reaction is used to pump two molecules of calcium from one side to the other side. So effectively, ATP is a highly efficient catalyst that performs a function while hydrolyzing ATP. It is a molecular assembly of which the function, pumping from one side to the other, is regulated through a chemical reaction. So that is what I mean when I say I would like to regulate our molecular assemblies by chemical reactions, because biology in this beautiful example does that so nicely. Why do we want to do that? Why would we want to mimic something that biology is much better than we? On the one hand, I think it will increase our understanding of biology. So that's an extremely complicated protein. How simple can we make such a pump? Like, does it need to be such a sophisticated piece of machinery? Can we lose a bit of efficiency? And then by losing efficiency, we can make our molecules simpler. So we could increase our understanding of biology. Also, when biology goes wrong, in the case of disease, we could increase our understanding of life. Like, what is actually principally, when is life and when is life chemistry? Where does chemistry transition to biology? And we could increase our understanding of maybe the origin of life. Like, where did it then come from? When did chemistry become biology? We can increase our fundamental understanding of supramolecular chemistry. How far can we push this field? Can we go beyond the static structures that we have? And as I said, can we start using pumps to, I don't know, purify water, or can we use it for other types of applications that require dynamic functions? And hopefully, I'm wondering, we can develop materials that have functions like living materials, or functions like the fact that it adapts to a change around it, the function that it self-heals, the way our skin self-heal, the fact that we can control it over different length scales, but also over space and over time. So these are the reasons why we are developing molecular self-assembly in a second toolbox, which would be molecular self-assembly regulated through chemical reactions. How do we now do that? The first thing that we identified, and as Sabine was already mentioning, I've worked on this in my PhD as well, with the vision of Jan van Ness and Rink Ilkema. Very early on had this vision, okay, we need to start introducing that in supramolecular chemistry. And what we did over there is we worked with ester-based reaction cycles. And we recognized, well, that was a very nice proof of principle. We need a versatile reaction cycle that we can use on many types of assemblies, but other people can also easily have access to. And relatively early, we found that this is a easy reaction cycle. It's absolutely, well, obviously I'm biased, but I think it's absolutely fantastic because it's straightforward to work with, it's scalable, and it's extremely versatile. Let me talk you through. We have a precursor molecule over here, and the precursor molecule is like our ATPase. It's in its low resting state, let's say. It's not activated or anything. And it is now the catalyst in a reaction cycle that is going to convert a fuel, carbodiamides, into its waste at the expense of water. Sorry, my coffee machine is switching off. It's going to take a couple of seconds. This catalyst, this precursor, reacts with our fuel, carbodiamides, and it's condensing agent, so it rapidly forms the O-acylurea, and then that converts into an anhydride. So we have a condensing agent that removes one water molecule from our precursor, converts it into the anhydrides. We do this in water, so this anhydride has a finite lifetime ranging in the 20 seconds, tunable, over the course of 20 seconds, and it hydrolyzes back again to our precursor. It's very important. This hydrolysis back is very important. So we activate, we go back. We activate, we go back. So we have a catalyst that is activated, goes back, and it's effectively just hydrolyzing carbodiamides. That's all we do. Just like the ATPase is hydrolyzing ATP, we are hydrolyzing carbodiamides. That gives us an active state, and that active state has a certain half-life with which we can play then. Can we couple that to a function of our supramolecular material? My dream is, of course, to build an ATP pump or something like that, but we're very far from that. Why do I like it? It works in water, that's nice, so we can get closer to biology, pH from 5 to 7, 5 to 50 degrees. It's versatile in the precursor, and I think that's actually the most important part. You can put any kind of dicarboxylate on here. R can be peptides, proteins, DNA assemblies, nanoparticles, etc. And the half-life of the anhydride, we can tune that now in the range of tens of seconds, all the way to hundreds of minutes. And that's nice because it allows you to tune for how long a molecule is active. Do you want very fast active responses where assemblies respond very quickly? Or would you like to build up something very slowly where molecules have time to diffuse and perform their function? It's now a reaction cycle that is relatively widely applied by other groups as well. And I draw over here, the precursor over here, and all the typical active states that other people and us have worked with. where depending on which nucleophile you choose, you get either a symmetric anhydride, a cyclic anhydride, activated esters, and other pyridine-based activated esters. So this is widely applied by us, but also by other research groups right now. But all of them have the same idea. We're activating a precursor molecule at the expense of a carbodiamide. And as I said, the nice part over here is depending on what type of active ester you choose or what type of anhydride you choose, you can tune this half-life in the range of tens of seconds up to several hours, in this case, 10,000 minutes. So we have a wide range over which we can tune the activity of our reaction cycle. Now, just as an idea, okay, that's nice. We know our reaction cycle. How do we understand all of these rate constants? What do we typically do? Usually when we come with a new precursor, we design those to form assemblies. So the first thing we usually do is have a look at the kinetics, because I think if you want to fundamentally understand what are my molecules doing, how am I controlling their function, you need to understand how long this molecule is active for, how long does it take to activate it, how long does it take to deactivate. So we usually go to HPLC NMR measurements to measure concentrations throughout the reaction cycle. And then we use kinetic models to predict concentrations. And this is what that typically looks like. So the markers over here would be data set from our HPLC. We apply a batch of fuel. We look at the emergence of our anhydride product, and then we look at the decay as a function of time. And then we apply a kinetic model from which we can calculate the conversion of the EDC. We can calculate the conversion of our, the building up of our anhydrides, and then the decay of the product. And these models are powerful because they allow you then to calculate, okay, what would happen if you add more fuel, if you add less fuel, if you change pH, if you change temperature, et cetera, et cetera. So they help us a lot to predict, okay, how can I affect, for example, material properties? If I want something that lasts for 2 hours or for 10 minutes, a model helps you to apply that. I think this is a fantastic study by Fabian. He came to me at some point and said, well, I think what we're doing is all wrong because we're measuring this anhydride and we're doing that on the HPLC. And when we do that on the HPLC, actually this anhydride keeps on hydrolyzing. We're injecting it, we're removing it from the carbodiamide fuel source, and it keeps on hydrolyzing. So we're not measuring the actual concentration, we're measuring the actual concentration minus whatever has been hydrolyzed on the column. So he invented a quenching method. The quenching method that I thought was pretty smart is just applying benzylamine. On the one hand, benzylamine immediately reacts with all the anhydrides and OSurea. That's the only downside. So you don't know if it's the OSurea or the anhydride in this solution, but it also quenches the activation immediately by raising the pH and converting your carbodiamide source. So now we have much more accurate prediction of our concentrations. And the other nice thing is that you can quench your solution and then just store it, just like biochemists like to quench their solution, store it on ice. We can do that now. And we can now hook that up to pipetting robots as well. So that is fantastic because that basically means you can load up a solution and then load up the fuel solution. You let the robot just mix it all together. You install a 96 well plate over here, and then you let the robot just pipette the whole night. It's just pipetting, quenching solution, storing it, quenching solution, storing it. Next day, you load your 96 well plates in HPLC, and you just measure the whole well plate through. And that gives you an idea that it starts automating your experiments, but also facilitates the pathway to machine learning and predicting experiments, where you can basically start letting algorithms predict, okay, what are the concentrations that are coming on, and what are the parameters that I should choose if I want more anhydride, if I want less anhydride, if I want faster reaction cycles, etc. Okay, so now let's regulate some assemblies with that. So far, we've only been talking about this reaction cycle, but what can we do with them? So what I'm going to talk about is a little bit about fibers, where we are regulating the behavior of fibers, as well as compartments. So where we're basically a newer direction that we're interested in is, can we actually change the behavior of molecular compartments that could serve as synthetic life at some point. So let's dive into those molecular assemblies based on fibers. In this study, what we did is we coupled this reaction cycle to the formation of peptide-based fibers. And what we typically do in such a study is we screen how can you now design these fibers. So what we do is our active group over here, that's the catalyst, let's say, that's the molecule that gets activated and deactivated, and you now see it in a precursor form. And in the precursor form, it has those two negative charges. And these two negative charges prevent this molecule from assembling, because on the other side, we've got a relatively hydrophobic phenylaniline isoleucine, so it would like to assemble, but it cannot because of those negative charges. And then what we do is we apply fuel, and honestly, And the first setup we do is after we synthesize those peptides, we do what I call a webcam assay. It's not the most fantastic assay. You just take your peptides, you add a bit of fuel, you put it in front of a webcam, and you just have a look what happens. And here what we see is that if we take molecule 1D, for example, only has one isoleucine, put it in front of the webcam, we apply fuel, and actually nothing happens. We see no evidence of fiber formation because the solution remains transparent. And obviously then the rest of the studies also start. We have a look at the beta sheet content. We have a look if there's an increase in signals that are indicative of self-assemblies, and we get nothing. That means that the self-assembling domain over here, the phenylalanine and the isoleucine, is not sufficient to induce self-assembly. Even if this is activated into its anhydride state, it doesn't self-assemble, probably because the driving forces for self-assembly are not sufficient here. We increase by adding another isoleucine. We do exactly the same webcam assay, and we immediately see the Coacervate solution. Moreover, if we now take this gel vial, we would invert it, or if we take this vial, sorry, we invert it. We see that this solution remains into a gel state. It's a very clear indication for us that something changed, the viscosity has changed. We have an assembly in there that scatters light and probably has something to do with fibers because it seems to form a gel network. And gel network for us is a good indication that there's fibers in there that change the viscosity of the medium. And then indeed, if we do all the assays that tell you there's a gel rheology and THT assays, they say like, well, there's fibers in there, cryo-TEM show there are plenty of fibers in there. Look at the fibers there, and these little dots over there, they're all background noise. It's ice that's actually formed during the freezing of our sample. But the fibers over here is what we're looking for. And the funny thing is, of course, this is a chemically fueled process. So if we now add our 25 millimoles of fuel, we get those fibers, but they are transient because at some point the system runs out of fuel. So if we don't keep on supplying fuel, the system runs out of chemical energy and we go back to our original precursor state. We go back to everything is hydrolyzed. We now have a solution with our precursor that went through several thousand cycles, and there is no more fibers because there's no more fuel left. So we have converted all the fuel. You can push this idea a little bit further. You can add another isoleucine, and then we found like, well, if you do that, you're actually overdoing it. There's now so many hydrophobic interactions and hydrogen bonding interactions that these negative charges over here are not sufficient to disrupt the assemblies. So without fuel, we already have a hydrogel. You can add more fuel to it, and the hydrogel gets a bit stiffer, but that's it. You don't go to another morphology. So what take-home message from this study is, of course, that you need to be careful with how you design your precursor, but even with very simple FIID peptides, you can now induce that into fibers reversibly. Essentially, what we did is we are dealing with energy landscapes, and we're populating states in the energy landscapes by applying chemical fuel. And in order to do that, the chemical fuel basically pushes up molecules higher up in the energy landscape. We're populating a state higher in the energy landscape. But in this case, the fuel, if you push it up, there's no state over there that represents a self-assembled state. So we push in fuel, but then we come again, we activate the anhydride and it comes down again, but there's no assembly process that is favored over here. In this case, what we have is we push in chemical fuel. We now do see the assemblies, but upon deactivation, molecules come back again, and we're back in this precursor state. And finally, if you overdo this and you put in too many isoleucines, then the precursor itself already assembles. So you can apply fuel to this, you can push this higher in the energy landscape, but it will not disassemble or anything. It will change the morphology of the assemblies. So what we show in such a study is that the molecular design over here regulates how our chemical energy is used. You can have no assembly at all. You can have chemically fueled or dissipative self-assembly, or you can have the fact that the precursor already assembles, and then you're ending up in a completely different energy landscape. And this is then what that looks like. If you add different amounts of chemical fuel, you see that you convert it into a hydrogel. This is, of course, a time lapse, so things go a little bit faster. And as a function of time, these hydrogels are collapsing. And you can set when this happens, depending on the amount of chemical fuel you add. The more fuel you add, the longer this non-equilibrium state is populated, and for the longer you have these fibers floating around. In the background, you see similar fibers. It's not exactly those fibers, but the image with the confocal microscope, not the cryo-TEM, but the confocal microscope, where you get also an idea of those dynamics. You see these fibers that are moving, and they are moving a bit more than you would expect than in equilibrium. There's dynamics going on, assembly and disassembly dynamics, but also when you rupture these delts, they tend to be more dynamic than classical in equilibrium dynamic fibers. In another study based on FMOC AVD, so it's another molecular design, similar in the sense that it also forms fibers. We found an interesting behavior that I'm very excited about to explore further, is that if we compare FMOC AVD, which can form fibers, to acetyl FAVD, which does not self-assemble. So we did our proper studies. There's no molecular assemblies at all. It doesn't form anything. It just remains perfectly in solution in its activated state. We find that we get very different kinetics, and that's super surprising, of course, because you have AVD, amino acid. Technically, the chemistry is very similar. The only difference is that it can assemble. And when it does assemble, when it is able to assemble FMOC AVD, we find that the kinetics, the EDC consumption, so the fuel consumption, is 5 times faster. And the hydrolysis reaction is also twice as fast. So both the activation reaction and the deactivation reaction is faster. So what do we have now? The assembly seems to somehow catalyze the reaction cycle. And I find this very interesting, because it means that on the one hand, the reaction cycle is affecting the fact that it assembles. So the assembly is affected by the reaction cycle. And the opposite is also true. The assembly is affecting its reaction cycle. So you have this kind of reciprocal coupling idea, where chemical reaction cycle is affecting assembly and the assembly is affecting the chemical reaction cycle. And I think this is something that we need to explore further, where we can basically force molecules to have to go into the assembly before they get deactivated. And that's also what biology is much better at than us, in making sure that the reaction cycle can actually not proceed until we have an assembly step, and only then can it go to the next step. Then you can really force molecules to have to do that function before they go to the next step. How does this work? We found out that the pKa of these assemblies is very strongly dependent on whether, or the pKa of these peptides, I should say, is very strongly dependent on whether they're in an assembly or not. So when our peptide is able to form an assembly, it changes the local environment of that peptide. That changes the pKa, shifts the pKa up, and therefore the carboxylic acids are actually partially protonated when they're in the assembly. So first of all, we have a mixture of precursor and product in these assemblies. And the precursor that is in the assemblies is partially protonated. And that accelerates the reaction, the activation reaction. So that explains why, if there are assemblies present, precursors co-assembled, and that changes, accelerates the activation part. We don't fully understand why the hydrolysis reaction, so when it's in its anhydride state, why this is also accelerated. We think because the peptide may be a bit more rigid, but we don't fully understand that yet. But we're very excited about being able to regulate chemical reactions with the assembly. So with that, I would like to move on a little bit to how we are using this chemical reaction cycle, not to regulate the behavior of fibers, but to create and regulate the behavior of compartments. So what we're looking for is a precursor molecule that upon activation becomes a product molecule, and that then phase separates or assembles into various compartments. I'm excited about those because they can serve as a model for cells, like how we can create cells out of thin air, or not out of solutions by applying chemical energy, but also how these cells decay over time again, so that they're energy dependent. So we are now populating non-equilibrium states that are representing compartments, entities. And I think that's exciting, where you have basically this entity is made out of a solution and it only has a finite lifetime before it will decay, unless it is constantly converting chemical energy, very similar to what cells are doing. On a conceptual level, it's very far away from an actual cell, of course. Compartments we are playing with are oil droplets, simply because of their simplicity. It's literally pentanoic acid. We add chemical energy and we get an oil droplet, and the oil droplet is only around for as long as there's chemical energy present. I think that's very simple and very origin of lifey. Even though an oil droplet is of course not a cell yet, very far from a cell, but it is, you're creating non-equilibrium compartments with very simple building blocks. But more recently, we started digging into Coacervate-based droplets, where Coacervate means a cationic peptide glues together a polyanion. And we've played around with vesicle and blockopolymer micelles. I would like to focus on those chemically fueled emulsions, where in this case, emulsions, I mean the droplets, the oil droplets of the Coacervate droplets. And why I'm excited about those is because they represent a key step towards the synthesis of life. We have now a compartment that is a non-equilibrium compartment that constantly need to convert chemical energy in order to stay alive. So we're getting closer and closer to the definition of life, a self-sustaining non-equilibrium system that undergoes Darwinian evolution. So we have no idea how to implement the Darwinian evolution yet. We're very far from that, but we're getting closer towards that self-sustaining part. They can serve as a model for membranous organelles, where membranous organelles are a hot type, well, very hot topic of research in the last 5 to 10 years, where it was identified that we have organelles, classical organelles that have a membrane, as well as organelles without a membrane. And what holds these organelles together is not a membrane, but it is the fact that it's phase separated. We have liquid-liquid phase separation within our cells happening right now. And the liquid-liquid phase separation is also tiny droplets floating around and also regulated through energy converting processes. So we're excited about studying those better, especially with these kind of model systems. And I think they can serve as a model for protocells as well, the origin of life. So how do we design that? So what we did is we use our chemical reaction cycle, of course, the one that we like so much. And as you can see over here, maybe a bit tiny, we have our reactive building block over here. So our catalytic building block, I should say. That's catalytic converting EDC, activating into the anhydride, and then back again. And now we need to use that to affect the phase separating behavior of this entire molecule. That's the reason why we designed this whole molecule around it. It's a peptide that is hydrophobic, phenylalanine, arginine, glycine, arginine, glycine, arginine, glycine. And the RG repeat is a repeat that we often see in phase separation in membranous organelles as well. So it doesn't come out of nowhere. We found it in biology is relevant. The reason why it's relevant is because RG, that repeat, loves to stick to RNA and DNA or other polyanions. So this RG repeat can stick to RNA and DNA and then can glue it together into what is called a Coacervate. So that's a droplet. It's basically high concentration peptide, high concentration RNA stuck together with this kind of cationic peptides. And what we did is we take this RG units and we cancel the fact that they can glue together RNA with these aspartic acids. They basically shield these cations and therefore make it less sticky to RNA. It is only +1 because it's zwitterionic, 3 + 2 - it's only +1. So it's not a very good at sticking together RNA. And then when we condense it into our anhydride, now it becomes suddenly 3+ molecule that sticks to RNA like crazy. This likes RNA, it floats around, swims until it interacts with RNA. These interacted RNA strands can now glue together into this Coacervate droplet. And that Coacervate droplet is then a non-equilibrium state that can only be accessed if you chemically fuel this. But what is exciting about this is that this is basically the influx of material is dependent on this chemical reaction, whereas the outflux of droplet material is dependent on the deactivation reaction. So if this deactivation reaction is very fast, it happens in the droplets. We've shown that this happens in the droplets. Then we start losing materials and we're losing this kind of active state. So activation is happening outside, deactivation is happening inside because there's plenty of water within these droplets. This is what we do. As I said, we love our webcam. So we put a sample in front of the webcam, just have a look at it, apply our fuel. In 2 minutes, the sample gets very turbid. And in 18 minutes, all the turbidity is gone. This is fantastic. This makes me super happy when we get a result like this. I have to say this was after screening, I think 15 peptides. So it's not the first hit. We screened several peptides. This was a hit. And then what we do is we measure the turbidity, the black markers over here as a function of time. And this corresponds nicely. Within 20 minutes, our sample is clear again. We also measure the concentration of product with the assays that I mentioned earlier. And these are the red markers. And what gets me very excited is the fact that they are perfectly overlapping. So there's very strong correlation between the concentrations we have and the turbidity. And that's a very good indication that molecules, when they get deactivated, quickly leave droplets. So the droplet population very accurately follows how much product you have around. So it's good because that means that this system is dynamic. Whenever molecules are in surplus, then we get a lot of droplets. Whenever molecules are getting killed, then we lose droplet material. And then what we do is we trap everything in a microfluidic bubble. This is the microfluidic bubble, microfluidic droplet. It's a bit confusing. We have a water droplet inside of a perfluorinated oil droplet, but within the water droplet, we have all the ingredients for our chemical reaction cycle. So what you will see now is little droplets that are appearing if I start running this movie. So what you see is a lot of immediately this turbidity comes up. These are the RNA strands that we're looking at. And the RNA strands, they are glued together by the peptides. So that's why you saw a lot of tiny droplets in the beginning. And what you see is these tiny droplets, they grow and they grow and they grow, not by taking up more material, but actually by eating up other droplets. So they're fusing together into multiple droplets until all the chemical fuel is done. And then it's game over. Even for the biggest droplet that is around, it also breaks up. So I'm excited about this because it basically means that out of nowhere, we have all of these droplets. They're competing for fuel. They're all fighting for fuel. There's limited fuel around. Some of them get large by eating up their neighbors. And then in the end, towards the end of the cycle, we're losing fuel, we're losing droplet material, and the droplets start shrinking and start dying out until even the last droplet has also disappeared. So we have this emergence, we've got growth through fusion, we have a process called vacuole formation, I'll explain that later, and decay. And we can track each individual droplet over its entire lifetime. Already here, let me explain the details over here. So what we see is this fusion event. Here's an example of that. It happens very quickly. So over the course of seconds, we see droplets fuse. These droplets are liquid, of course. This is a good indication for us that they are indeed liquid. And they have a very low viscosity or very low interfacial surface tension as well. So they like to fuse together. If we now go to this, all of these droplets, and we start tracking these droplets, we take actually the last droplets we had in the cycle. So the 1, 2, 3 last droplets. Let's call them blue, green, and orange. Now we can go back in time because we can use tracking software to go back in time and we can actually see where this is a blue droplet, where did you come from? So at some point you ate your neighbor over here. So the next time frame, those two are eaten and then we end up with that droplet. We can do the same for the green. That's actually the result of 2 fusion events. The orange is the result of 4 fusion events, etc., etc. You can go even further back in time and see actually all the blue came from here, all the orange came from here, the green came from here. And here you can learn already a couple of things. The droplets don't tend to travel very far for their food. They eat their neighbors, but they don't go all the way to the other side in order to travel around. And the other thing that I think is cool is that you can now see that there was a red population at some point and a purple population. And the red and the purple population have died off. So they were dead before others were dead. So the droplets have an individual lifetime. But just like real cells, they die at different time points. And that means that there is competition. At some point, the blue one, just one, did not allow for the red one to get enough nutrients and therefore it just died off. I think these are the ingredients to start thinking about Darwinian evolution, meaning that we can now start thinking about how can we extend the lifetime of one droplet and how can we make sure that there is competition and this competition becomes beneficial for that droplet. But then we also need, of course, the fact that you transfer this information to your next generation of droplets. This is something that we're very far away from. When fuel runs low, we see this weird behavior. This is typically non-equilibrium behavior. We don't see this in equilibrium. We look at this droplet over here, for example, which we call vacuole formation. Towards the end of its cycle, it is dissolving, obviously, the droplet disappears, but it doesn't dissolve as I expected. It doesn't like stuff leaves from it. Actually, the inside of the droplet is decaying. It's as if you're going from a bowling ball to a soccer ball. So the inside is now decayed and they still have a shell left. And we think, again, this has to do with the fact that there is still more material from outside is being added onto. Activation is still happening. There's still fuel present. So from the outside, we still get material, but not from the inside. In the inside, there's no fuel. So the inside is decaying faster than the outside is actually decaying. So it's yet again a behavior that is driven by influxes and outfluxes, which are driven by a chemical reaction cycle. So it's a very non-equilibrium-like behavior. And then the last spectacle, and this is actually a soccer ball, or this is what they're actually hollow inside. And then the last spectacle is when almost all fuel is depleted. We are like the inside is decaying. Followed, I think, this droplet over here. What we see is the droplet doesn't dissolve in the end as I would expect. It just crumbles apart into tiny droplets. I like to call them baby droplets, of course, because I'm thinking in terms of how can we think of the next generation, getting all these tiny baby droplets, hundreds of baby droplets coming from one droplet. And you see already the concentration of baby droplets starts increasing because all of these droplets are decaying. So suddenly you get all of these tiny fragment droplets. And the fragment droplets, they survive for tens of seconds, maybe a minute if you're lucky, but that's it. And then it's game over and everything is gone. So what's going on through my mind is how can we save those daughter droplets? Can we start playing Darwinian evolution tricks if we now fuel exactly at the right time? So we fuel, we starve our population, then we fuel again. Can we save these baby droplets from death and start playing evolution games with those? Finally, if you continuously fuel it, so when the fuel concentration is kept constant by constantly applying chemical fuel, we have the impression that the droplet size remains constant. But this is very preliminary data that we're playing around with. So what you see is the defocus and the change of the movie all the time is because we're applying a new batch of fuel. We're applying fuel, And we're doing this now for an hour, whereas the other population would have already died off, of course. we see that the droplet size remains relatively controlled. So we have the impression that this is a method of controlling droplet size. And this is where we come in with this models for biology, for memberless organelles. This is also a method by which biology can control its organelles, which is still a mystery. How does biology control sizes within the cell? As I said, this is an example of such an memberless organelles, the P granule. The P granule is also regulated through chemical reaction cycles and energy converting processes. And in this case, what the authors found in actually one of the first memberless organelles papers, they found that yes, you can control where these droplets dissolve and where the droplets appear by applying chemical gradients of proteins. Okay, I'm having a look at the time. Okay. Another compartment we are playing with is oil droplets. And the critical difference here is not the fact that they're oil versus coacervates, or complex coacervates. It's the fact that in this case, the activation and the deactivation both occur outside. So we find a different behavior in chemically fueled oil droplets. And this is partly because the activation happens outside. Like the previous example, the fuel prefers to stay outside, so therefore it happens outside. But in this case, there is no water within such a droplet. And therefore, because there's no water within such a droplet, the activation can only occur outside because water is of course required for hydrolysis. That's what those droplets look like. These are oil droplets floating around. What we find is a very weird behavior. And the weird behavior in this case is that the droplets, they grow. It's a function of time. Actually, they don't grow, they ripen. And ripening in this context means we have a lot of tiny droplets that come together somehow. The droplet material comes together into one large droplet, but the coming together does not occur through fusion, as in the previous example. It occurs through ripening. And the ripening is effectively that tiny droplets are thermodynamically not favored, whereas what the system wants, or what thermodynamics wants, is one very large droplet. This happens in oil water emulsions. This happens all the time in our vinaigrettes. We have oil and water, olive oil and vinegar, and the olive oil within your vinegar also tends to get larger and larger droplets, partially because of fusion together and partially because of Ostwald ripening. Now, fusion doesn't happen in this system. We have looked at these droplets for hours and hours and hours. We don't see any fusion, but we see ripening through Ostwald ripening. Now, that's not too surprising. It's a thermodynamic behavior. What is surprising is how quickly it happens. So Ostwald ripening is a very slow process, and we see extremely fast ripening. If we sustain our population of droplets homogeneously by constantly applying fuel, we see that these droplets grow very quickly. Moreover, if we now make droplets with different half-lives, so we change this reactive module through phthalic anhydrides, through linear anhydrides, cyclic anhydrides, and NHS esters, we change how fast this reaction happens, extremely fast, fast, intermediate, and slow. And that changes the deactivation rate, so how fast this reaction cycle is operating, changes how fast our droplets are growing. So over here, we plot the ripening rate, so how fast the droplets grow or ripen, and how fast the deactivation, and it scales nicely over orders and orders of magnitude. We get this relation. So there's a very clear, this is a very clear indication that a behavior we observe is regulated through chemical reaction kinetics. And indeed, for our fastest ripener, we see over the course of minutes, we get this very big droplets. So it's a signature of an active behavior. It is regulated through the chemistry and not through thermodynamics, or at least a combination of activation and thermodynamics. And then we start digging into this mechanism. We teamed up with a theoretical physicist. They came up with models for this. And basically, what fits extremely well is the following, is that Ostwald ripening is driven by a diffusive flux from small droplets to big droplets. Small droplets, as I said, are thermodynamically not favored, they have a higher Laplace pressure. Because of that, there's a small concentration gradient from a small droplet to a big droplet, meaning there's more droplet material around this small droplet than there. And if you would now deactivate molecules along this concentration gradient, what happens is you're actually removing molecules from that gradient. And now the question is which droplet is going to supply those molecules? Is it the big droplet that is thermodynamically more favored, or the small droplet? And the answer is, of course, it's more likely to come out of the small droplet. So the deactivation reaction is sucking effectively small droplets dry, whereas the activation reaction is doing the exact opposite. It's now creating molecules between these droplets. And where can these droplets go? They can go to the small droplet or the big droplet. If they go to the small droplet, they have to diffuse up a concentration gradient, but is not favored. They diffuse down. So the deactivation kills small droplets, the activation grows big droplets. And this is the reason why we can accelerate oswald ripening. And this works for both if we apply one batch of fuel, as well as if we continuously add our fuel. And if you now team up with these theoretical physicists, they can do calculations and they can calculate what you would expect for regular oswald ripening and how much it is accelerated by. So this is our droplet growth, our volume. It's growing quickly. Blue is what you would expect for classical oswald ripening. And as I said earlier, it's a slow process. This doesn't happen very fast. If you now take a fast reaction cycle, you can accelerate it by almost 2 orders of magnitude. You can make it 100 times faster simply by making it an active process. So you're wasting chemical energy, but thereby you are accelerating a property that is already there in the thermodynamics. So you're making that 100 times faster. You can correlate that. We now take the rescaled deactivation rate, so how fast our reaction is, and we can now calculate how fast we're accelerating oswald ripening. And as I said, you can go all the way up to 2 orders of magnitude faster than you would expect for this classical behavior. Why do we care about this oswald ripening? Because it gives you control over droplet growth. Imagine you're a cell and you would like to build up a couple of organelles real quickly. If you need to wait for oswald ripening, that's a slow process. You need to wait for molecules to fuse from one side to the other side. If you could accelerate that somehow, not the diffusion, but if you can change the concentration gradients by applying chemical gradients, you can control how fast droplets are growing. So you can control how fast your organelles are growing. And you can do that simply by activating outside and deactivating the outside as well, resulting in this concentration gradient or playing around with these concentration gradients. Similarly, if you do the opposite, you're activating outside, you're deactivating inside, you can control droplet size. So these are the kind of ideas that we're playing with. How can chemical reactions regulate the size of compartments or the behavior of compartments, the growth of compartments or their size? One last example, going a little bit into applications, is that because activation and deactivation is happening both outside, we found a little bit surprising decay profile of our anhydride. And the surprising decay profile is if you have anhydrides that don't form droplets. So C4 or C6 have a 2 tiny hydrophobic tail to form droplets. We get classical first order decay kinetics. It's nothing surprising over here. If you add fuel, you grow your anhydride and then it decays with first order kinetics. If you now take C8, for example, that can phase separate, that forms droplets, you suddenly transition to 0 order kinetics. If you do C10, which can also phase separate, but even have a lower solubility, you get again 0 order kinetics, but much slower. So you can control the rate of this anhydride hydrolysis by the solubility, and you can control the rate constant also by making droplets. So we think, okay, what's going on over here? It's actually relatively straightforward. The deactivation reaction is a first order reaction that is determined by the concentration of your droplet molecules. And the concentration of droplet molecules is regulated by the droplet because it's equal to the solubility. The droplets do not allow more materials to be outside, cannot change the solubility. And every molecule that is above the solubility is protected in such a droplet because there's no hydrolysis within the droplet. So that means the concentration of droplet material in solution remains perfectly stable until the last droplet has disappeared, and then we transition to first order kinetics. In other words, the droplets are protecting the anhydride from hydrolyzing, changing the reaction order from first order to 0th order, which is now determined by the concentration of droplet material, which is determined by the concentration, by the solubility of the droplet material. You can see that very clearly if you transition from C8 to C10, the solubility drops by an order of magnitude, and indeed this line becomes an order of magnitude flatter. The hydrolysis rate decreases an order of magnitude. We skip that. We were then thinking, can we apply this to deliver hydrophobic drugs with 0 order kinetics? So what happens now if we take this emulsion, we take these droplets, in fact, we ignore the EDC, we ignore the activation reaction, we just take the oil, sonicate it, add our drug, load it into these droplets, load the drug into a hydrogel, and then apply a buffer on top, and then measure the release of our hydrophobic drug into this hydrogel. And this works fantastically. If you don't have the oil in there, you get first order release. It's extremely fast. If you have the droplets in there, you get 0 order release of all of your hydrophobic drug that is controllable by the lifetime of the emulsion. You add more oil, then the lifetime of the emulsion is longer too, and then the release rate is slower as well. So we're playing around with that as well. We're thinking of this as an anti-inflammatory and antimicrobial delivery agent, for example, in a wound dressing where you have an emulsion trapped in a gel, so that's called an emulgel. And this emulgel is constantly releasing hydrophobic drugs over the course of 24 hours, 48 hours. You can set it by the concentration of your anhydride that you applied. Okay, I think with that, I would like to wrap up. Yeah, I think I've been talking for long enough. What have we learned? We are developing molecular self-assembly. And in molecular self-assembly 2.0, we have molecular self-assembly that are regulated through chemical reaction cycles. So the reaction cycle is a cycle of activation and deactivation. It's effectively a catalytic reaction cycle that is converting what we call a chemical fuel into waste. And the assembly can only happen at the expense of chemical fuel. That costs you energy. Obviously, that is not beneficial. You need to now invest energy, but you get something in return. And what I've shown you that you can get in return is a mechanism to control size, a mechanism to control growth, a method to store information in a dynamic reaction network. I didn't talk about that yet. It's currently under review. I had to skip that because of time. And a method to generate and erase information, and a method to generate or to create and decay compartments. And the compartment is something we're really playing around with and looking forward to methods of making these evolve as a function of time, or evolve preferably through Darwinian evolution, as they are competing with competition or with other droplets. What we're playing with that I did not talk about today is using the high precision molecular engineering of DNA origami combined with the high precision of our reaction cycle. So we're playing also around with DNA origami strands in order to change the folding of molecules at an expense of chemical energy, as well as the emergence of information by oligomerizing, just like DNA and RNA does, oligomerizing molecules so that we can, information suddenly emerges at the expense of chemical energy. With that, I would like to thank all the fantastic people in my team, students as well as my alumni. And I would like to thank you all for listening to me, and I would welcome any questions.